So just remember, at least by Thursday, sign up for the courtesy access of Connect, right? If you want, pay the $24, pay the extra $25. We did find out today it's $25 for the loose leaf to get shipped to you in 2 to 6 days, and they are pretty quick about it. Or, you could stick with the ebook. Students in the past have said if you buy the $24 and then later it's only like $50 to add the ebook. So the other thing is if you choose just the Connect assignments because you're low on funds right now or something, right, you could add the ebook later. The ebook, the actual textbook is on reserve at the library as well. Our library is still currently located in Building 10, which is right behind this one, kind of, across from the Student Life. There is a brand new library. They screwed up the electrical. That's why they're not in there yet. It's what we call the lowest bidder, y'all. One of my friends, I had to find her. Like, her office, she moved over, and I was looking for her. She's my embedded librarian. She usually works with me, and she actually usually embedded into my course and does stuff for honor sections and different things that I've done. And I went to go find her, and I was like, wow, this place is kind of scary. She's like, oh, yeah, it's a whole lot of scary. Which is really unfortunate. I mean, we have this big, brand new building, and basically nobody's in it. A lot of the books they've moved in out of the trailers that they were in. But I have it on reserve. So like if you're studying on campus or something like that, although I'll tell you, I haven't looked up the library's hours this summer. They have their webpage embedded in our Canvas course, which is nice. You don't have to search Delgados' website for them. So we should actually find the answer to that question on their page. All right, ask a librarian. Their home. For the first 2 weeks, yes. After that, you'd have to pay for it. Yeah. Does anybody see the hours and I'm just not seeing it? They have their search thing, my account, forms, special services. Huh? Maybe about us. About us. Has their contact information. Oh, it is there. Monday through Thursday, 8 to 8. There we go. 8 to 8. Yeah, they're still not doing Saturdays. I don't know, we have Saturday classes, but during the regular semester, I should say. In the summer, we even shut down on Fridays. Okay, so answers that question. So back to this. So that's all the stuff about the book, right? Here, let me make it look like it does for you guys. Okay, so notice that the attempts are unlimited. This assignment, if you click on it, you'll see it's actually 0 points. And it's actually old biology questions I think I picked. But what I did is I picked like an animation question. I picked one where you have to drag and drop. And the ones that require different what's called plugins on your computers, right? So people have run into problems in the past trying to use things like Safari because Safari doesn't support Flash, and a lot of these types of things run on Flash. So if you just do this assignment, like if you're having trouble, right, try and do this assignment. That might be the issue, is that, oh, well, I don't have what I need, and it should hopefully tell you. And that's what I say here, what you need. So let's see if this computer I'm using will let me do this assignment. So when you click on it, right, it says Start Activities, right? It asks you a few questions. I'm just going to click Random Things. Then I'm going to Submit. And because it was random, I got them all wrong. It tells me the right answers. All right, but then the next part is an animation. So things that could be wrong is, you don't have the correct player to play this, right? Or sometimes, you know, it depends on where you're at, how good your internet connection is, right? That's another thing to take into consideration, especially when you're trying to play videos, right? And, you know, downloading and enough time for these things to go. So it looks like this one will go.
During glycolysis, glucose is
Not a
broken down
problem.
to
These little
pyruvate.
spots, it's going to ask us questions, right? I wasn't listening. What was it, mitochondria?
A 2-carbon fragment of pyruvate is used.
You can usually fast forward.
A 2-carbon fragment of pyruvate is
There it
used
is.
to form acetyl CoA. The acetyl CoA enters the
Apparently
Krebs
won't let
cycle,
me skip
which
over it.
occurs in the mitochondrion. During the conversion of pyruvate to acetyl CoA, carbon dioxide, CO2,
So I'm gonna be
is
bad.
produced, and a molecule of
And I'm just going
NH
to skip over that one. And this is the other type, like drag and drop, right? So like if you can't do it, it's probably because you don't, like I said, have the player. Now that would be at the bottom, right? And then there. Anybody know what this is? This is glycolysis, right? Breakdown of glucose. And when you don't have oxygen in our bodies, we make lactose, which is not good. It's acidic. All right, what's the last one? Probably another. Oh, so just another animation where it's not the multiple step animation like the other one, but it has the questions at the bottom. But again, if you're having trouble
during
playing
glycolysis
something,
and the tricarboxylic.
right, check and see if you've got the right plugins. Does that make sense to you guys? So some people, you know, I mean, you guys get all kinds of different devices now. I guess I can't even keep up, right? Oh my God. You know, I have this assignment so you can just play around, right? And it doesn't stress you out, right? Because none of this is for points. So I'm going to save and exit. When you save and exit, it doesn't record your score, right? You have to actually submit to record a score, right? So, you know, I didn't even finish it, but I'm going to submit it. And so then I get my results, right? Feedback on what I did. And then I can, this one's that big long one, so then you can skip to the next one, right? And I got it right. That one I damn well better get right. I know that one. All right, I didn't even bother answering that one. Or the other ones that went along with it, right? I just skipped right to the end. So I didn't get any points, but it's not worth any points, so no big deal, right? Although it says 100%. It's backwards. All right, so I forget how to navigate from your guys' view. Software check. I guess because I'm in student view. As an instructor, it's giving me issues. Okay, so once we finish chapter 1 tonight, one of the good things to do right before Thursday's class, hopefully, is to take and do this chapter 1 as if it was a quiz, right? And I'm going to warn you guys, a couple of these questions, I couldn't edit out some of the scientists that they have in here. So if it isn't one we didn't talk about in class, don't worry about it, right? Because again, I would have edited it out if I could, but I can't, right? But I have handpicked all these questions in Connect, right? All these assignments here that you see. And you'll notice that right now, I just went ahead and set the due date for like the end of our summer session. I know you guys are diligent students. I don't think I need to babysit you and plug in individual dates for each one of these, right? As we complete the material, I expect you guys to, as I said, take this as if it's a quiz. Close your book. Close your notes. See how much you really know after you've studied. And then you can do it again. Notice they all say unlimited. And so you could even do them again before the final if you want, if you found that they were helpful when you took the test, right, to review. Again, don't memorize these questions. I'm not going to ask you, obviously, the same questions. The question is, do you know the material, right, that we've covered in the class? So there's a thing called Learn Smart. Some people may have stumbled upon it in Connect, right here. I haven't set this up. And the reason is it's quite time consuming, and I cannot edit out questions that you guys don't want to deal with because they're not related to what we're covering in the class. So it's set up to cover everything in the chapter, right? So I could pare it down, but I can't select out the ones I don't cover. Does that make sense to you guys? It doesn't, this particular program, I don't have that ability. So I've tried in the past to use it, but students get very annoyed when they get questions that they're not going to get tested on. They think it's a waste of their time. And I somewhat agree. And then it's also a waste of my time too. So I would advise, don't mess around with that, except maybe when we get to immunology, because we basically cover everything in those chapters. Right. And actually, I think, did I put one? I've tried it several times. Sometimes I have a bonus one there that's a Learn Smart, to try and help students that are struggling. But I don't think I did it last semester. So, any questions on Connect? Right. So the unfortunate thing is right, I have it linked to Canvas, but you actually have to go into Connect, right? It's paired and actually brings you to a separate website. So right now the grades are in this separate one, but I'm going to try and learn this week how to actually pair the grade book too. So hopefully all of it will transfer over, which will make it easier on me grading time and probably help you guys better keep track of your progress if everything's all in one spot too. So that's something we had with Blackboard, but they just finally made it possible for Canvas. The publisher has to do that. It's not anything we have to do. So we've been waiting on them. Okay, so back to our course. So I see that at least, like when I come into my main page, it tells me what you guys have been doing. And we have 11 students in this class, and I have 11 submissions. So that's pretty awesome. That means everybody's introduced themselves. I haven't got a chance to look at them yet. I spent the day at the zoo with eight-year-olds. I might need therapy now. That was brutal. I don't know why I agreed to these things. They were like, we did not go near the gorillas. That wasn't on purpose, though. But yeah, no one fell in anywhere. But they ran everywhere. So I didn't get to read these, but I will soon, and I look forward to it. So at this point, everybody's done at least their initial post, but don't forget you have to reply to 2 of your classmates. So now everybody has posted. If you haven't, as you guys were working on it, replied to 2 of your classmates, make sure you do that. So that way you get all of your points. You get all 10 of your points. Chemicals to use to clean. This is actually for Module 2, hence it says Module 2. So if you went ahead and did it ahead of time, that's no big deal, right? And the same thing with the discussion board. That's actually Module 3, Immunology. I just have everything open already for you guys all for the whole semester. And those dates, again, are just kind of guesstimations. We may change them. And so that was another date that somebody asked me is, well, when are we going to take our first test? So we had Audrey, it's D, right? Audrey, okay. You weren't here. Do you have a laptop or tablet? Okay. Which one? Tablet, okay. Okay. So what we were thinking about doing instead of doing Scantrons, which I think all of us hate, is actually, I already have all the tests in Canvas. They're computerized. But you obviously need some type of computer device to take it. But the computer lab closes at 6 during the summer. I could probably ask her to stay open later, but then we have to set that date ahead of time too, right? And it doesn't really give us much flexibility. But there's only 11 of us. There's plenty of room in this classroom, and the Wi-Fi has been pretty good. So I want to do a test run on Thursday. I want you guys to bring in whatever it is you think you're going to try and use for the test. And we're going to do a little quiz, the agreement quiz. But in order to do testing, you have to have a special program on your tablet or your laptop, and it's called Lockdown Browser. And so it's free. And I just added it. Let me go to my student view for you guys. So that's one of your things for homework before Thursday. Here's my student view. So under our modules, right, under exams, download Lockdown Browser. So this is what we use for testing. And this tells you about testing. And if you scroll down, right here is the link. So this is if you have a computer, if you're using a Mac or some other PC. If you're using a tablet, I know for, let's just click on it. Right, so before you install, you could figure out if you could do it. I know for iPads, in the iTunes store, there's Respondus Lockdown Browser. That I know for sure because there's a setting in this for me to allow tablets, iPads. I don't know about the other devices for you guys. You have the Windows, oh, the tablet, so you would probably be able to download whatever this is. Yeah, okay. So that's what I want you guys to do is I want you guys to download this onto whatever you're going to use for testing and bring whatever you're going to use for testing on Thursday. And I'm going to have you guys turn on your laptops, get onto the Wi-Fi, and take a quick quiz on Thursday. Now, depending on how we move through the material, we could actually also finish the material on Thursday. But I don't necessarily want to give you guys a test on Tuesday, unless you want it. But we can't waste time in this class, right? So what I would do on Tuesday is start covering unit or module 2 material. And then Thursday, we would come in the following Thursday, take the test first thing on your devices, and then afterwards continue lecturing on module 2. Sound good? Anyone have an objection to that? Yes, absolutely. So there's a potential that we'll finish the material on Thursday. But instead of having the test on Tuesday, we'd have it the following Thursday. But that means that that Tuesday, we would start covering the next set of material that would be on the next test. Okay? And also give you guys an opportunity over the weekend, do your connects, right? Compile if you have any questions. We can do a short review even on that Tuesday. So plan for that Thursday. You guys need a date? Yeah, thank you. So you can write it on your calendars. But I definitely, download this program, right? And bring your device with you. And then Dylan, you don't have, but I said that you were going to use mine. Or okay. And if you do, if you don't, then you're welcome to use this because I won't need it while you guys are taking the test. And it's the Lockdown Browser will stop you guys from being able to do anything on your tablets or laptops except the test. So I don't have a problem letting you use it because you won't be able to do anything but the test. Any other questions? Nope, we're good? Okay, so let's get to the good stuff. All right. Okay. Let's see where we left off in lecture. Oh, one thing I wanted to point out. Audrey, did you watch the recordings from last time? No. Do you know where they are? Okay. 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 So just a review for anyone else in case you miss, right, class, but definitely contact me if you're going to miss or you have missed. So that way I can fill you in on anything that may have not been recorded, but as you can see, again, I'm already recording. So I actually even left up the old lectures from last semester, right? But right here under this module, what I'll keep doing is I'll keep adding them. And then the day one, which was all the, sign up for everything, was under day 1 right here, day 1 recording. So if you need to review any of that. What is this? Oh, this assignment is overdue because I'm in my student view. It's telling me I didn't do it. I never seen that symbol before. I was curious as to what it was. So.
Couldn't you still do it?
Yeah, you can still do it. Yeah, that's the other thing is when I set deadlines, those are guidelines. If for some reason something happened, right, and I haven't graded it yet, do it. Because once I grade it, I'm less likely to let you do it, right? But if I haven't, or if you're not sure, just do it, right? If you're not sure, just do it. And then that way, when I go to grade, right, you'll have something there. And you don't have to say, oh, well, I wasn't sure. Just do it. Right? Okay. Because stuff happens, right, to all of us, unfortunately. And I actually made my husband not turn off the internet and pack up all our stuff to move to the new house Thursday night so I could upload that lecture for you guys right away. So he was like pacing around the living room waiting for that lecture to upload. I was like, I didn't want to stay here Thursday night. I wanted to get home. It was uploading and I was packing my kitchen while I was uploading for you guys. So, all right. So, yeah, let's get to lecture. Let's get to the good stuff. So, last time we were talking about microbiology in general, right? And we talked about different classifications. And so let's actually see how much you guys remember. Let's try and fill in this table from memory. Don't look at your notes, right? So, what's a cell type? What are our 2 choices? Prokaryotes and eukaryotes. And what are bacteria? Prokaryotes. And archaea? Prokaryotes. And eukarya, why is it called that? Because they're eukaryotes. Okay. So cell organization we're talking about, is the organism a single cell, unicellular, or is it made up of multiple cells? So for bacteria, they're unicellular, right? A single cell is a single organism. What about archaea? Unicellular. And actually that's something that's linked to prokaryotes as well, right? Prokaryotes are all unicellular. Eukaryotes, on the other hand, can they be unicellular? Yes. Can they be multicellular? Absolutely. We're eukaryotes, right? We're made of eukaryotic cells. We're definitely multicellular. Cell walls containing peptidoglycan. Now, this type of cell wall is unique to a single domain on this list. Which one? Bacteria. So the answer for the rest of the guys is no. Does that mean they don't have cell walls? No, it just means they don't have cell walls made of peptidoglycan, a substance we're going to talk about soon. Membrane-bound organelles. This is a defining characteristic of a particular cell type. Which cell type has organelles? Eukaryotes. So the answer for the first 2 is no, and then yes. Where can you find bacteria? Oh, you name it, right? They're there. All environments. What about archaea? Right, you'll find them in all environments, but they tend to favor the extremes, right? Really hot, really cold, really salty. What about eukarya? Anything that's non-extreme, right? They don't like the extremes, like me, right? If it's too hot or too cold, they're like Goldilocks. They're like, nah, no thank you. Right? I like it just right. So and then we kind of touched a little bit on viruses, right? And remember, this is different classification scheme because they're not even cells, right? They're not considered living by most scientists. And we're going to spend a good amount of time talking about them in our next unit, in our next module. So we just slightly introduce them here. So then that brings us to good old Anthony von Leeuwenhoek here. He's a very curious individual. And he's the first to observe and describe microorganisms accurately. And the reason why there's dates here is just to give you guys an idea, other than the picture. This was a really long time ago, right? Microbiology's been around for a long time, thanks to very curious individuals like him. He was actually a drapery merchant, and so he was very interested in the fabrics in which he was purchasing. And so he made actually his own lenses. He ground down glass to make lenses to be able to observe in greater detail the fabrics that he was working with, right? To actually see the thread count, right, and things like that. But he didn't stop there, right? He had this ability to see in more detail, right? So why not look at more things, right? So he looked at things like pond water. He even looked at his own poop and was the first scientist to discover Giardia, a protozoa that can be found sometimes in your intestines, right? So he was definitely a very curious individual. And I was reading a text not too long ago and found out that these drawings, he didn't even do them. He hired an artist to do them. I was like, really? It's not that complicated. But that's why we know what he did, because what did he do? He saw it. How do we know he saw it? He wrote it down. He documented it, right? He wrote letters to the Royal Society in London, right? He hired an artist to draw what he saw. And in this one, y'all, it's not a semicolon. This is actually 2 little dots that he saw. And what happened here? So you have this little rod-shaped, and then they've got boop, 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 dotted. It moved. It moved. Right? So he was documenting that it moved. Now this is what they believe, he used to help support the objects that he was looking at in his lens. This isn't in fact what he had. They don't have that. So as we start seeing these things, right, that we never saw before, obviously we're going to have questions about it. We're going to try and explain things. That's just human nature. And there was this belief at one time in spontaneous generation. But it has been disproven as an erroneous idea. But what is this idea? What does spontaneous mean? All of a sudden, poof, right? Like magic. Generation meaning more, right? So, that apple juice that gets pushed to the back of the fridge and forgotten, you know what I'm talking about, right? Right. It's translucent. You can see through it, right? When you purchased it. If you find it in the back of the refrigerator, maybe a month later, right, after it's been opened and drank out of, and all of a sudden it's cloudy and there's stuff floating in it, who's going to drink it? Nobody? Why not? It's apple juice. It's gross. Why is it gross? What's gross about it? No. Well, then you can't enter into the conversation if you just don't like apple juice. But you said there's bacteria in it, right? And how do you know there's bacteria in it? Put their mouths on it. Yeah, I got news for you. Do that in my house. I put your name on the jug and it becomes yours. Yeah. But what if nobody put their mouth on it? Why? How did it get what does that cloudiness mean? It means that microorganisms grew, and a lot of them grew, right? To large, because we're talking about microscopic organisms. So you usually can't see them, right? But if they grow in large numbers in a liquid like apple juice, then you could see it as that cloudy mass, right? That says, oh, no, I'm going to pass on that one, right? So we know these things, right? Nowadays. They didn't necessarily know that back then. So they thought maybe just poof, apple juice could turn into microorganisms, right? We now know, right, it's microorganisms that came from somewhere. Like, so she said somebody put their mouth on it. But in my house, we don't do that, right? Unless it's yours, right? And we're not sharing. Okay. So could that happen in my house then? Yeah, it can. Because how else would microorganisms get in there other than putting your mouth on it? The air. The minute you open that juice container, right, you expose it to the air. So microorganisms can get in that way. Right? We don't live in a sterile world, do we? Certainly not. Right? So they're everywhere, as we said. They're everywhere. And if you've taken lab right now, she'll tell you, right? They're everywhere. We swabbed everything, everything grew. Right? Microorganisms everywhere. So this belief, right, there are several scientists, and again, this is an abbreviated list. We're not going to go over everybody's experiment in every little detail. But again, you can see this happened a long time ago. Sometimes I just can't even fathom 1626. I mean, it's 2016. Can't even imagine what life was like back then, or how long ago that was. Just mind-blowing. Right? So again, this is not a history class. We're not memorizing dates. The dates are just there to give you guys some perspective of how long ago this conversation happened. So, what did we say? We said living organisms can develop from non-living or decomposing matter. Right? So that apple juice. But we now know, right, it's not the apple juice that turned into the microorganisms. It's the fact that the microorganisms got into that apple juice container and started eating that food source, the apple juice, and grew to large numbers under those conditions. So Francisco Reedy, very simplistic experiment. Not dealing necessarily with microorganisms, but smaller organisms. So flies are, of course, attractive to food. So something like the meat that he placed in jars, the flies come along, and they do what? They lay their eggs. But can you see the eggs? Not unless you look very closely, right? You usually are not going to see them. Why is she putting her eggs there? It's a food source, right? So when they hatch, right, they have something to eat. So then you see what? Maggots, right? That's the larval stage of a fly. So they actually believed at one time that meat turned into maggots because they didn't observe, right, the flies laying their eggs. So he showed this, and he went a couple steps further even. He completely encased the meat. So the flies could not get to it at all. They couldn't smell it. They couldn't lay their eggs. And again, no maggots formed because, of course, no eggs were laid. The next one, he covered it with like gauze, right, or cheesecloth. So there's air that could get through. There's holes in it. The flies could sense the nearby food. So they laid their eggs on that mesh, and actually the maggots developed on the mesh. But again, not on the meat. The meat did not turn into maggots. It didn't spontaneously generate. So most people were like, oh, okay, I guess we didn't pay that much attention. But, that's not true for apple juice, right? There were no flies involved, and we're not talking about maggots, right? So can spontaneous generation be true for microorganisms? And we know it's not, right? The problem is, again, they're so tiny you don't see their presence until they reach those large numbers. So John Needham took a food source in a liquid form, what we refer to as a broth. He boiled it, and he left it open. And then, of course, growth happened. And the problem here is he what? He left it open. And we know microorganisms are found in the air, right? So they settled in the food and they grew. Why did he boil it? Right, to kill anything living in it already. But does that always kill all microorganisms? No, we now know today, right, that there are some organisms that are heat resistant, even to the level of boiling. So that freaks some people out when I say that, right? Because what do they tell us to do when they think our water's contaminated? Boil it. But notice I said how many organisms are heat resistant? Some. Do we have to worry about those in our water supply typically? No. Right. And the good news is that all the times in New Orleans that we've been told to boil our water, it was just precautionary, right? No contamination occurred, which is good, right? It's always good to err on the side of safety. But why do you have to wait so long to know? Right? Shouldn't we be able to know this stuff instantaneously? What are you waiting on? You're waiting for the microbiologists to take the samples and grow the bacteria. And the good news is that when they take the samples and put them on those petri dishes, right, with the food, and they come back, nothing grew. Yay. We didn't drink anybody's poo, which is good, because one time I didn't even find out about the advisory until after I had already done a lot of things with water. So, yeah. So, okay, the next guy, Lazaro, decides, okay, I'm going to boil it, but now I'm going to stopper it, right? So no growth happens until he opens it. And so you guys are on the other side. Okay? What would you argue here? Why did stoppering it stop it? It wasn't exposed to air. Okay. But do all organisms need air to grow? We know they don't. But maybe back then they were like, oh, well, the reason why it didn't grow is because you didn't let the air in. Right? So he's like, okay, I'll open it up. And then look, it grows. But again, they didn't know at that time that it was in the air, right? It was an air problem here, right? It's in the air. You can't stop it. And you gave it food. And so it's going to grow. So depending on how you look at this and how much knowledge you had, you might say that this supports spontaneous generation, right? Doesn't really help disprove it. So it wasn't until my friend Louis Pasteur, which, of course, he's named after one of the processes we use to kill microorganisms, right? Pasteurization is named after him and developed by him. If there's ever the such thing as going back in time, I want to go back in time and have a glass of wine with this man. I want to pick his brain. He is pretty impressive. His list is huge of things that he did in his lifetime that affect us even today. So he actually filtered air through cotton, right? And doing that, he was able to trap the microorganisms that we couldn't see in that cotton, and then place that into a sterile broth, a broth containing no microorganisms, and actually cause growth to happen. So he's saying, look, right? There's stuff in the air. But he didn't just stop there, right? Of course, he's got to deal with those people who are like, wow, you know, the reason why it doesn't grow is because there's no air, right? You've got to have air for growth. So he's like, okay, fine. I'll give you air, but I'm not going to give you direct air. I'm going to give you indirect air. This air is going to have to travel through these, and they weren't this elegant. This is, you know, a drawing. They weren't this beautiful. But they're referred to as the swan neck flasks, right? They're stretched out and curved like a swan's neck. And the purpose of that is indirect airflow. So air, right, unless it was literally forced by wind, it's not going to be able to travel all the way here with the dust particles still in it. So the dust particles get trapped in this bend, right? And those are what the microorganisms are attached to. So air gets in, but no growth happens, right? He boiled it, he made it sterile, no growth happens. And then he shows that if you do open it, right, microorganisms will get in, growth happens. And in fact, in a dust-free room, right, in those really super clean rooms that they have for making certain devices and such, you can leave flasks of broth, apple juice, glass of apple juice on the shelf forever. Nothing will grow, right, because there aren't any microorganisms in that air, right, super clean. So everybody's happy, right? Spontaneous generation isn't true, right? Microorganisms are in the air, right? If you keep them out and still allow the air in, you're not going to have any growth. Is everybody always happy when someone tells you, yep, that's the way it is? No, right? Most people will be like, show me. Or, I'm going to try and prove you wrong, right? So they repeated his experiment. And some people did not get the same results as him, so they're arguing with him. They're like, no. But the problem was, and why, when you read scientific papers today, why they will literally tell you what lot number of petri dishes they use sometimes, right, for their experiment, is that if someone is to replicate their experiment, they would have to do all the same conditions if they wanted to try to disprove what that scientist did, right? You'd have to use the same type of petri dishes, the same type of food, right? The same environmental conditions, temperature, all of that, right? Because all of those things would affect. So the problem is that the people who were repeating his experiment didn't use the same food source. They believe that he probably used yeast extracts. They probably used hay extracts. And hay, a lot of times, because, how do we make hay? We cut grass, we lay it in the field, and we let it dry out in the sun, right? In the dirt. And we ball it up. Microorganisms in the soil, right? When you're exposed to soil and stuff like that, a lot of times you can get what's called endospores. This is the dormant form of some bacteria. Can you kill them with boiling? No. So that's the problem, is they use a different food. So therefore, even though they boiled, they didn't achieve sterility, where when he did his experiment, he did achieve a sterile environment when he boiled his food source, because he didn't have anything in it that was heat resistant at that time. Others, unfortunately, probably had heat resistant organisms in it. So it wasn't really the final blow until John and Ferdinand got onto the picture. And these two guys did similar work at two different labs around the same time. So they're both given the credit. And they really helped demonstrate even further that the dust in the air is what carries the microorganisms. And as I said, if dust isn't there, right, if you have a room that's been HEPA filtered, and that stands for high efficiency particle air filters, then you could create a sterile air environment. They also provided evidence for the existence of exceptionally heat-resistant forms of bacteria, what we now call today as endospores. So the other problem we ran into back around those times is that when people got disease or were ill, they would believe sometimes the gods were punishing them, right? They told a lie, right? All different things, because they really just couldn't, they were trying to explain, right, what's going on. One of my high school English teachers freaked out when they read my essay because I had an atheist time in my life, and I said, God is just everybody's answer to their unanswered questions, right? But that's how we are, right? We want answers to our questions, and we do the best we can to answer them with the knowledge that we have and the beliefs that we have in our lives. And so the same thing for people back then. And you will deal with that even today. Like, so how many of you guys want to be nurses? Right? So, God bless you. For real. Right? Whether you believe or not. Because you're going to deal with all walks of life, all levels of education. Right? And it's an art form to really help people, because you're going to have different beliefs than your patients, right? And different knowledge base, and you are trying to help them, but sometimes that can be very difficult because, right, you're not on that same belief page or knowledge page. When I go into my doctor's offices, if they don't know me, I don't tell them that I'm a scientist, right? Because it's an interview the first time I meet them. If they can't explain it to me, right, how they should, the information that I need to know, then they don't need to be my doctor. Things like if they don't wash their hands before they touch me, I get up and I walk out. Right? Things like that. Then I let them off the hook, after a little while. I'm like, you don't have to, go through the whole rigmarole. But my allergist, when I went to go see him one time, he's like, oh yeah, I don't really need to explain this to you, right? He's like, but can I explain it to your boyfriend? I said, sure, go for it. Right? So, he really wanted to make sure, right, that I understood, or even my significant understood what was going on with me. Right? And that's really important. And it's not an easy task, right, to do. And so we now know today, right, most of us know that microorganisms can cause disease, right? And of course, scientists had to prove this, right? But it was not immediately obvious because, again, we're dealing with microscopic things, right? Things we can't see with our own eyes. We need special tools and special abilities to see these things and study them and understand them. So to establish that connection, right, dependent on us developing these techniques, right, to be able to work with microorganisms. The most widely used tool, a microscope, right, had to be developed and utilized. The ability to grow organisms, to be able to study them, right, to be able to create these unnatural food sources and petri dishes, right, named after Petri. And once it was established that we could actually start studying the organisms themselves, we could even go one step further and really ask that question, well, how is it that some people don't get sick even though they're exposed, right? And this led to my favorite area of microbiology, immunology, right? How our immune systems work, or don't work for that matter sometimes, with disease. And some of the really early scientists lost a whole lot of friends. I can tell you that when I was studying homeopathic medicine, one of the founders of homeopathic medicine literally gave the different remedies to his friends and observed what happened as he increased the dosage. Yeah. Sometimes it wasn't very good results. And the thing is, I don't think they were willing participants. You know what I'm saying? Like nowadays, if you want to be involved in medical research, you sign, and sometimes you even get paid to do it. And one student one time said something to me, and I was like, whoa, yeah, not ethical. Not ethical at all. But at one time they tested on military. And I don't know if you signed up for the military and you signed some waiver you weren't realizing you signed, right? But they don't do that anymore. At least they're not supposed to do that anymore, right? Unless, again, you signed a waiver and you know that you're going to be given something experimentally. and observe to see what happens, right? We don't normally do that to people, right? But do we do that to animals? Yeah. And unfortunately, to a certain extent, that is necessary for us to know the things we know today, right? So at one time, yes, unfortunately, people were experimented on without their knowledge. Robert Koch developed this series of tests that could be performed to help link a specific disease to a specific microorganism. And these are referred to as Koch's postulates. And so the first relationship he was able to pin down, and probably the bacteria's name comes from the disease name. Anthrax is caused by the bacteria we now know as Bacillus anthracis. So he used the criteria he developed from his teacher, but of course he gets the credit. So they're referred to as Koch's postulates. And they're still used today, really not to the extent they were used in the past. We use a lot more molecular testing, DNA testing nowadays, than whole organismal testing. And that's because for some organisms we have some limitations as it applies to these postulates. So how, right? How do you establish this link? Well, first of all, he had to acquire the microorganism from a diseased organism, right? So he would take, say, a mouse that had been exposed, had anthrax. He would take a sample, observe that he had microorganisms present, and then we'd use a special technique that we're going to do in lab this week. It's called the streak plate method. And it's where he took the microorganisms, he spread them out on a petri dish on a solid surface. And the goal was to separate cells out from each other so that these growths, these little dots, these what we call isolated colonies, hopefully are a single organism, right? One species. Because when you spread it out, hopefully you spread them out so much so, right, that cluster of growth that you see is just from one organism multiplying millions of times. And then he would take that, right, so he would take just one of these colonies and inject it into a healthy mouse and observe and see if the same initial disease happens. But he wouldn't stop there. He'd go one step further again and make sure that he could get back that same organism that he started with. So can you do this for all infections? Can everything be grown in artificial culture? No, right? Mycobacterium leprae, when I did research in leprosy, we don't grow it in petri dishes. They grow it in armadillos and nude mouse footpads, right? You have to have an organism for it to grow. It won't grow in artificial culture. We can't provide all the essential things that it needs without a living organism. So that's a problem, right? Can mice get all the diseases we get? No. Human immunodeficiency virus is called that for a reason, because mainly it affects humans. Although rhesus monkeys are similar enough to us that they can actually use them to study that virus. Right. Can all organisms grow on their own without their other buddies? No. And there's some pretty amazing technology, I'll tell you, that's out there where they can grow cells on these little chips and be able to grow multiple organisms together, but yet analyze them separately. I haven't gotten to delve into that too much, but I'm looking forward to that workshop in 2 weeks. Find out about that stuff. So it's really amazing, advances in technology that we made to be able to, advance the study of microorganisms and in everything on the micro scale, right? Teeny tiny things. So some individuals can be infected by a pathogen, yet not develop the disease too, right? Some people are exposed and they don't get it. And some become chronic carriers where they're sick and they don't know it and they're continually shedding this. So how does this observation affect Koch's postulates? Right? It creates a problem for us, huh? How might the postulates be modified to account for the existence of chronic carriers? What changes could we make? So they don't look sick, right? But they have the infection. Right? We can do other types of testing, right? Maybe genetic testing and different things, right? So, we have to take these variables into consideration. And that's also why when they do these types of tests, they don't just inject one mouse, right? They're going to do like 10 or 20 or several, right? Because you're going to sometimes run into ones that their immune system is going to take care of it, and they're not going to get sick, right? There is a tribe in Africa that's genetically immune to HIV. That virus must attach to receptors on your cells to gain entry into those cells. This particular group of people, genetically, they're so genetically different that the virus cannot attach to their receptors on their cells. So they won the genetic lottery, right? We can't steal this, make it into a therapy, right? It's their genetics. So we had to develop techniques to be able to do this study, right? So his led to the advent of agar. Agar is a polysaccharide derived from marine algae. The cool thing about this sugar complex is that it will stay solid at relatively high temperatures. You literally have to boil it to liquefy it. So we can put it in an incubator at 37 degrees Celsius, 98.6 degrees Fahrenheit, and it won't melt. Gelatin, on the other hand, which is a protein, which we sometimes use to solidify things like shrimp molds, other than Jell-O, right? But can you put that in an incubator at that? No, you can't even leave it out at room temperature. It starts melting, right? And the problem with that is, I don't know, how many of you guys like gelatin? No, I do, right? I like Jell-O. It's a good protein source. It's great for your nails. Lots of bacteria like to eat it too. So they would produce enzymes and digest it and liquefy it anyways, right? So, and not everybody likes potato, right? That was one of the ways that they would, they'd use potato slices to have a solid surface to grow microorganisms. So with the advent of agar, where we could add this polysaccharide to other foods and create a solid surface, was a huge advancement in the study of microbiology. These little things are kind of trivial to some of us that have been using it for a long time. But when you think about it, you're like, wow, when they figured this out, they changed the world. Literally changed the world with, something this simple. The Petri dish, that design of that dish is all based on Louis' work, right? Because if you were to look at that dish, you have the bottom, right? And you have the lid. And anybody notice something about the bottom and the lid? They're loose. They don't twist on or anything like that. They don't seal shut. It actually creates indirect airflow, right? The lid is a little bit bigger than the bottom. And there's a reason for that. Indirect airflow. So air can get in, but the dust doesn't. Nutrient broth and nutrient agar. Again, we're using food, whether it's in water, which makes it a broth, a liquid suspension, Or if we add that food to the agar, then we could create a solid surface, whether it be in a petri dish, or sometimes we'll even just put it in a tube, and we'll lay that tube on a slant as it solidifies, as it cools, and they'll give us a nice slanted surface inside the tube. And I was going to bring some of that stuff over for you guys to see tonight, and I got distracted. But I'll bring some non-contaminated media to class Thursday so you guys can see what microbiologists use. And then the ability to isolate, right? Separate out individual organisms to be able to do that streak plate method. So our contributors, these are the important guys you need to know what they did. So Anthony von Leeuwenhoek, what do you think of when I say him? Right, first to accurately describe and look at, right? And the important thing there is, again, he documented it, right? Maybe other people looked at microorganisms before him. He might not be the very first, but he was definitely the very first documented case that we've been able to recover. Reedy, what did he help us with? What do we say? Bzz, bzz. Yeah, the flies in the meat, right? And Pasteur, yeah, the swan neck flask and the importance of the indirect airflow, helping disprove spontaneous generation. Tyndall and Cohen, they furthered Pasteur's work and helped us know about what type of organisms. The heat resistant, the endospores, yep. Koch, of course, is Koch's postulate, so what do we think of with him? We think of diseases, right? What organisms cause what specific diseases? We haven't talked about Lister yet, but there's something you may have in your bathroom at home that's actually named after him. Listerine. He didn't invent Listerine, it was named after him. It's an antiseptic, right, that we use to rinse our mouths. And he was one of the first to realize that with surgery, you pose certain risks. So he was one of the first to actually clean the instruments and the skin of the individuals that he operated on and had less post-operative infections because of it. And then Carl Woese, we mentioned briefly, he came up with a system of classification that we use today. What division of classification is thanks to him? The domains, yep. So in addition, remember I said Pasteur's list is long. This is, again, just a few of his major accomplishments. So with other individuals, Pasteur discovered that incubation of cultures for long intervals between transfers cause pathogens, organisms we know to cause disease, to lose their ability to cause disease. What do we call this? No. We call this attenuated. And you may have heard this term before, because they use these type of organisms to vaccinate you, right? So when they say the vaccine is attenuated, it means that the organisms have been altered or weakened in some way. For some of them, it's just growing them in artificial culture for a long time. I refer to this as the fat and lazy syndrome. Because think about this. If you just keep feeding somebody and they don't have to work for it, they're going to sit on the couch and become fat and lazy, right? And so that's basically what you're doing for microorganisms when you grow them in artificial culture. You're just going, here, food, here, food, here, food. You don't have to work for it. Here you go. I'm giving it to you for free. But when they're pathogenic, do they have to fight for that food? Yeah, because they're in another organism. They're having to fight with other organisms there or that organism's defense system. So they've got to really work for it. They have abilities that they have to keep up. I always think of WALL-E. You guys seen the movie WALL-E? Right? Everybody out in space gets fat and lazy because they don't exercise, and the whole problem with space, no gravity, decreases bone density. You have no pressure. So similar thing happens to microorganisms. So again, Pasteur and other people were able to use some of their studies to develop vaccines, right, for chicken cholera, for anthrax, and even rabies. So vaccinations against various childhood diseases have contributed to the entry of women, particularly mothers, into the full-time workforce. So why would that be true? Well, if your child died, you could still go to work though, right? The problem is if we vaccinate, then the kids don't get what? Sick, so therefore mom can go to work instead of staying home with the sick kids. When I was a kid, your parents purposely exposed you to chicken pox, right? When someone in the neighborhood had chicken pox, you went over and you played with them. And I've even heard that some parties, chicken pox parties, the kids were allowed to share lollipops because it's respiratorily transmitted, but they wanted to really make sure everybody got sick. You're looking at me like I'm crazy, right? Oh yeah. The whole purpose was to get it over with before you went to school, right? So I got mine when I was 4 years old from my chickenpox party. My little sister must have been a very clean player because she did not get chickenpox until she was in high school and missed 2 weeks of school. Not to mention the complications go up the older you are, and the fear gets insane if you're pregnant, right? What's the good news nowadays? Do you have to worry about chickenpox? Anyone got kids? Why don't we have to worry about chickenpox? Vaccination. So there is the possibility we may even reach eradication of chickenpox. Wouldn't that be awesome? If you're anybody like me, I still remember chickenpox, and I still have my scars from picking those suckers off. And I had them everywhere, literally. It was horrible. My mom duct taped mittens on my hands. It was traumatic. My son will hopefully never go through that, because in 1995, they developed the vaccine. But if you're vaccinated, does that mean you will never get shingles? No, because what is shingles? It's chickenpox reactivated, right? When it hides in your sensory nerves and it goes, boop, hey, I'm here again. The problem with that is that your immune system is what keeps it under control most of the time. And when you're stressed, right, and you don't take care of yourself, or as we age and our immune system wanes, right, then you get shingles. So what do we do to help prevent that? We vaccinate. Guess what it is? The chickenpox vaccine, just in a different dose. Right? We're boosting your immunity. We're going, hey, don't forget, this guy is in there. Keep him under check. Keep him under lock and key. Don't let him out. Because in this case, it's not in your skin. It's in your sensory nerve ganglion. And it's extremely painful from what I've been told. Right? And so usually when younger people get it, right, that's a sure sign that you are not leading the right lifestyle. You are way too stressed, right? Or you got some immunological issues you need to look into. And then, of course, if you're a professor here, we all walk around and say you have herpes because it's a herpes virus. Most people have herpes anyways. Okay, so he also demonstrated alcohol fermentations and other fermentations were the result of microbial activity. And he also really helped out the wine industry, right? Because sometimes they were producing wine and it was just horrible, right? It was like, oh my God. But it was getting contaminated with other microorganisms that don't produce what we like, right? That's how he developed the process of pasteurization, where you could heat it up and kill off the bad guys, right? The ones that we didn't want. And, we use this nowadays. Most of your juices, the reason why they don't have to be refrigerated is because they've been pasteurized, which means that most of the microorganisms that were present naturally in the environment have been reduced to a substantially reduced amount, so much so that you can put it on the shelf. But then once you open it, remember, you expose it to more organisms, and so refrigeration helps to what? slow down the growth, right? So there are some places, especially in Europe, you can buy milk on the shelves. It's a different type of pasteurization, right? And because of that, the milk is different too, and most Americans don't like the taste of it because we didn't grow up drinking it, right? We grew up drinking refrigerated, differently pasteurized milk, not ultra pasteurized milk. So it's never really taken on in the United States. In Europe, it's very, very common. And so sometimes, we'll get people that come from other countries, especially in New Orleans, right? They love it, and they're like, I don't understand. Why is your milk in the freezer case? Or in the icebox, and I'm like, it is. They're like, where's the other milk? I'm like, we don't like that milk. You won't find that milk here. And vice versa, right? Americans go overseas and you're like, where's the milk? And they send you to the shelves and you're like, oh no, I'm not doing that. That doesn't look great. That can't be possible, right? It can, but it's different. So Lister, as I said, right, he provided indirect evidence, right, that microorganisms cause disease. And he developed a system designed to help prevent wounds from getting infected, especially surgical wounds. So he actually, treated the dressings with antiseptics and the instruments, and his patients probably really loved him for that. Right? Did you need something? Okay, hold on just a second, because we're getting ready to take a break anyways.